Greetings, Bentley alumni, students, family, faculty, staff, and friends. Thank you once again for joining us for today's virtual event. I'm Emily Williams, Director of Alumni and Family Engagement. Before we get started, as always, I have some light housekeeping. After this session, we'll be sending out an email with a recording and a survey to gather your feedback. Definitely feel free to suggest future programs of interest to you. Thank you to those of you who submitted questions in advance. Zoe will be addressing several of the pre-submitted questions on this webinar. If you have a question arise as you're listening, please put it into the Q&A box and it will be considered for response during the question and answer session after the presentation. Lastly, if you need technical assistance, please use the chat box to speak with us directly. My colleague Bethany will be responding to your questions as best she can. And now today's featured speaker is Bentley University lecturer Zoë Wagner. Zoe, PhD, is a pharmaceutical scientist and bioengineer with a background in development of novel protein-based nanoparticle therapeutics for targeted treatment of solid tumors. Her research broadly seeks to understand how underlying biological structure and function can be used in the development of novel pharmaceutical interventions. Wagner's current research focuses on quantifying pharmaceutical innovation, natural product drug discovery, and pedagogies of science, technology, engineering, and mathematics, or STEM, education. We had Zoe with us as one of our very first virtual events back in March of 2020, and now she's back for part two. So without further ado, please join me in virtually welcoming back Zoe.
Hello, thank you so much. Good afternoon, everyone. Thanks so much for joining us today to talk a little bit more about the SARS-CoV-2 virus and vaccines. So, as was said, I am a pharmaceutical scientist here at Bentley University, and I am going to start, as I did last time, with just a few disclaimers. First of all, I am not a medical doctor. Nothing that I say today should be taken as medical advice. If you have any questions about how the vaccine may impact you or your loved ones, or what schedule you should be on for your booster shot, I suggest you reach out to your physician. We can talk about what the vaccines do and why the booster might be effective, but if you have any sort of personal medical questions about that, please reach out to your physician. Secondly, while we certainly know a lot more about this virus than we did in spring of 2020 when I first talked about it, there is still a lot we don't know. And this is particularly true when it comes to questions about immunity, antibody levels, and protection against some of these viral variants that we're hearing more about. I am going to try to summarize the state of knowledge today, but please keep in mind a few things. First of all, there is not a single right answer for a lot of the questions that you may have today. There are a lot of complexities, there's a lot of nuance here that are really important to consider. And I recognize that that can be frustrating, right? We want things to be clear. We want things to be black and white, A or B. But unfortunately, that may not be possible, right? We are going to continue to gather more information and make appropriate decisions. But that is what we should be doing, right? Keep in mind that at the very core of scientific inquiry and at the very core of this process of knowledge acquisition and novel investigation that we are undergoing right now, the very core of that is this need to adapt, to change our understanding and to change our actions based on new information. We are constantly revising and adding and remodeling our understanding of this virus and the vaccines. So the information that I talk about today may shift, it may change, it may be updated today or next week or next month, and that is okay. That is exactly how this process is supposed to work. And in fact, that shows that this process is working, all right? So we're going to take in new information as it comes, and if appropriate, we will change actions and update guidelines. So let's begin with the basics. What is SARS-CoV-2? SARS-CoV-2 is the virus that causes COVID-19. I am today going to be talking a little bit more about how the virus causes infection in our body and how our immune system responds to that infection, and then how we use vaccines to sort of take advantage of this natural immune response. And so talk a little bit about the vaccines that are currently available for COVID-19 and what are we talking about when we talk about antibody levels? Why might that matter? What are these variants of interest? Why might they matter? How can we sort of begin to create a holistic understanding of why we want to vaccinate, what the vaccines are doing, and how they're helping to protect us? So viruses are pretty unique in that they can only replicate inside a host. And this is one of the key features that differentiates a virus from bacteria. If you leave bacteria, such as E. coli, on a surface, if you give them some food and some oxygen, the bacteria will reproduce, they will divide. If you do the same thing to a virus, the virus is unable to reproduce. It will just sort of dry out eventually. The virus requires a host in order to reproduce. So what the virus does, if it gains entry into a host cell, is that they sort of hijack the cell's, the host cellular machinery and turn the cell into a viral production factory. So how does this happen? How does a virus reproduce using a host cell? To begin to understand that, I would like everybody to think of that sort of quintessential image of the coronavirus, right? And so you have that sort of, it's usually depicted for whatever reason as sort of a grayish surface coating on the virus with all of these spike proteins sticking out from the viral envelope or the shell of the virus. And those spike proteins are what the coronavirus is sort of best known for. And it's how it got its name, right? Those spike proteins provide or are depicted as a halo or corona around the virus itself. So this spike protein is going to stick out from the envelope or the shell of the virus. And that's on the outside. On the inside of the virus, there is the RNA genome. The genome is all of the genetic material that is required to create a new fully functional virus. This is much the same way that our own human genome comprises all of the DNA that is necessary to constitute the structures and functions of the human body. SARS-CoV-2 is an RNA virus, which means that it stores all of its genetic information as RNA rather than DNA. RNA is structurally pretty similar to DNA. It is single-stranded rather than the double strand that we're used to seeing for DNA. But the function of RNA is to provide a template for the production of protein. So in humans, our genetic information is stored as DNA, and that DNA is converted into RNA, and the RNA is then converted into functional protein, and protein is the sort of actionable element in our bodies. Similarly, with SARS-CoV-2, the genetic information is stored as RNA, and this RNA, when it encounters a host, is converted to viral protein. The way that the virus gains entry into our cells is that following an exposure, the viral protein, the viral spike protein, binds to receptors on the host cell, on human cells, and this receptor is called ACE2. And the spike protein binds to the ACE2 receptor using a region called the receptor binding domain, or RBD. And once bound to the ACE2 receptor, the virus is sort of locked in place. You have the spike protein and the ACE2 receptor, and they're sort of locked together. And a second human protein that has the somewhat unfortunate name of TMPRSS2 is then going to come in, and it's just going to snip a little piece of the spike protein. And by making this cut, it allows the spike protein to undergo this really dramatic change in shape. And the spike protein is going to sort of unfold. And as it unfolds, it's going to pull the virus together with the host cell. So it's going to pull these two things together and actually allow the virus in the cell to merge. So now the virus has merged with the host cell. And once that happens, the viral RNA is released inside the host cell. So the viral RNA is now inside the human cell. Once inside the human cell, the viral RNA is going to be converted using that same host cellular machinery into viral proteins. And the production of more copies of viral RNA and viral protein begins. Because remember, our cells are naturally doing this. all the time. Our cells are naturally taking DNA, converting it into RNA, and converting that RNA into protein. So when the virus enters into the host cell and injects that viral RNA is going to be picked up by our own cellular machinery and converted into viral protein. So now we have these cells in our bodies that are churning out copies of the viral RNA and viral proteins. The protein and RNA are then repackaged back into more viruses, into more virions, and these viral particles leave the host cell and disperse to find other cells to infect. So you now have the host, in this case, us humans, the host cells are churning out these viral particles. What does the body do, right? What does the body do in response to this? And our immune system is what protects against and fights against infection, be that from viruses, from bacteria, from fungi, any other foreign invader, anything that I'm going to call a pathogen. And generally, we think of there being two parts of the immune system, innate immunity and adaptive immunity. The innate immune system is sort of our first line of defense. The cells that are part of the innate immune system are not specific to a particular pathogen, but rather they recognize characteristics of pathogens, characteristics of these foreign substances that cause disease. And the response, once a cell of the innate immune system recognizes a pathogen, is generally to consume or phagocytize it, quite literally to engulf it and break it down. And there are several types of cells that are part of our innate immune system that are responsible for this process of recognizing pathogens and then destroying them by this phagocytosis, by engulfing them and breaking them down. So that's the innate immune system. It is fast to respond and it is non-specific, right? It is going to sort of seek out anything that is viewed as foreign and try to destroy it by breaking it down. And that is a very important role of the innate immune system. But the innate immune system also plays an important role in communicating to this other part of our immune system, the adaptive immune system. So what the innate immune system will do is say, hey, this thing, this particular foreign invader, this particular pathogen, this is bad, right? We need to remember this so that we are able to respond more quickly next time. So the adaptive immune system works to create a specific response to a given pathogen. And to ensure that there is specific recognition of these pathogens, we have specialized cells that are called dendritic cells. And these dendritic cells identify regions of pathogens that act as identifiers, effectively little flags that say, this thing is bad. And once these specific areas have been identified, and we call these sort of specific flags antigens. Once they've been identified, we have another cell in the immune system that's called a B lymphocyte. And B lymphocytes begin making antibodies. And these antibodies are specific to that particular antigen, right? So we have the innate immune system that is recognizing something is foreign, right? This thing should not be here. And it is identifying specific elements of that pathogen that act as flags. And these flags are called antigens. So those antigens, again, are flagging something as being foreign. And B lymphocytes create antibodies specifically for that antigen. Antibodies are something we've all probably been hearing quite a bit about. They are Y-shaped proteins that circulate in our bodies. They are highly specific. So a single antibody is going to be highly specific for a single antigen. And these antibodies are going to circulate in your blood for some amount of time. And if they come into contact with their specific antigen, they're going to rapidly bind to it. And the binding of antibodies to the antigen signals to the rest of the immune system that a foreign entity has been identified and it must be killed or neutralized. So even after antibodies are no longer circulating, we also have what are called memory cells, which remember specific antigens and can rapidly go into action if the body encounters the same antigen again. These memory cells rapidly stimulate the production of more antibodies so that a familiar threat can be neutralized. So in a SARS-CoV-2 infection, the part of the virus that is recognized by the immune system is the spike protein, that protein that sticks off of the surface of the virus. So the antibodies that are produced in response to infection by SARS-CoV-2 are specific to a portion of the viral spike protein. Additionally, the binding of antibodies to the antigen can prevent the antigen from attacking cells. So in the case of the SARS-CoV-2, if there are existing antibodies against the virus, those antibodies are going to bind to the spike protein. When the antibodies are bound to the spike protein, that's going to prevent any interaction between the spike protein and the ACE2 receptor. And so it may prevent the virus from being able to effectively enter into host cells. This process of antibody production, however, is going to take some time, on the order of a few weeks, in order to fully ramp up. So those are sort of our two parts of the immune system: our fast initial non-specific response of the innate immune system of identifying and then engulfing and destroying foreign invaders, but then also the portion of the adaptive immune system, which is slower, but much more specific and focuses on antibody-mediated responses to these foreign invaders. So this immune response is what we take advantage of when we make vaccines. Vaccines function by exposing the body to a version of a pathogen that is incapable of causing disease, but is recognized by the immune system as foreign. So when exposed to a vaccine, the body recognizes the active component as foreign and prepares an immune response. And this includes the production of antibodies and memory cells. This means that if you are exposed later, after vaccination, to a disease-causing version of the pathogen, your immune system is already trained to recognize it. You already have circulating antibodies against that particular pathogen. And so your body is able to rapidly clear the pathogen from your system. This means that if you are vaccinated against a particular disease and then subsequently exposed to it, your body may be able to clear the pathogen from your system before it can take hold and cause an infection, or your body is able to have a dramatically increased response, so the infection caused by the pathogen is much less severe than it would otherwise have been. And we have a few different types of vaccines that we are sort of used to receiving. An inactivated vaccine contains a killed version of the pathogen that causes disease. So an example of this is the polio vaccine or some versions of the flu vaccine. They inject a heat-killed or heat-inactivated version of the polio or flu virus. The virus is fully incapacitated and so incapable of infecting cells. However, it is still, structurally, it is still recognized as foreign by the immune system. And so the immune system kicks into action, producing antibodies against the polio virus or the flu virus. So if you are ever exposed to an actually active version of these viruses, your immune system is ready to sort of rapidly destroy the threat. We also use what are called attenuated viruses. These are weakened versions of disease-causing pathogens. The pathogen is not strong enough to cause active infection, but it does still produce a robust immune response and a robust antibody response. And these include the MMR vaccine and vaccines for chickenpox and smallpox. So a type of vaccine that's definitely worth talking about, specifically for coronavirus, is the mRNA vaccine. Both Moderna and Pfizer are mRNA vaccines. If you remember back to when we were talking about the structure of the SARS-CoV-2 virus, we said that the genetic information for the virus was stored as RNA, and this RNA is converted into functional viral protein. And this viral protein, specifically the spike protein, is what the immune system recognizes when mounting an immune response. So in an mRNA vaccine, we take advantage of this process. The vaccine consists of the RNA necessary to make a portion of the SARS-CoV-2 spike protein. This RNA, for just a portion of the spike protein, is packaged inside a lipid particle and injected intramuscularly into the arm. Once injected, the lipid particle is going to allow the RNA to be released inside cells And from there, the cellular machinery is going to take over, and it is going to begin translating that RNA into the viral spike protein. So what happens is that without ever having been actually exposed to the CoV-2 virus or to the full viral genome, the cells are now making pieces of the CoV-2 spike protein. And this portion of the spike protein is recognized as foreign, and the immune system gets to work producing antibodies against that spike protein. It's important to note that the mRNA vaccines do not interact with DNA in any way. Rather, the information in the vaccine is used to create pieces of the spike protein, which then trigger an immune response. Generally, the first dose of the mRNA vaccine trains the immune system to recognize the viral spike protein, and the second dose provides and ensures that sort of robust antibody response. And that ensures that if there's a subsequent exposure to the coronavirus, the body's immune system already recognizes the viral spike protein and already has antibodies in place to respond quickly to the threat. We also have the Johnson & Johnson vaccine, which is a vector vaccine. The J&J vaccine uses a delivery vehicle called a vector to deliver a piece of the SARS-CoV-2 genetic material. So the vector, the delivery vehicle, in the case of the J&J vaccine, is called an adenovirus. And adenoviruses are very common. They cause things like a cold or pink eye. So very common, but very minor infections. And in this vaccine, we've taken the adenovirus and we've altered it. So it cannot make you sick. It can't replicate, but what it can still do is bind to host cells and enter into the host cell to deliver genetic information. So we've taken just sort of the outside of this altered adenovirus, and inside the virus, we have placed a piece of the SARS-CoV-2 genome inside. In this case, specifically, a piece of the genome that codes for the viral spike protein. So when administered, the adenovirus binds to host cells and injects pieces of the genetic material from the virus. The host cell then begins making viral protein, and the immune system responds in the exact same way that we just described for the mRNA vaccine in terms of creating a robust antibody response. So while Moderna and Pfizer are the first mRNA vaccines to hit the market, I think it's really important to note that research into mRNA vaccines has been going on for decades. This is not new technology. This is something that researchers and clinicians have been studying and optimizing for years. And the pandemic certainly spurred and increased the rate of development. But these vaccines were built off of a significant and solid foundation, right? And so there was already really robust research that was in place at the beginning of the pandemic that allowed us to rapidly pivot and develop effective vaccines against COVID-19. So one of the questions that people often have about these vaccines have to do with side effects, right? And so following vaccination, people are likely to have side effects that include things like pain at the injection site. It might get red, it might swell a little bit, and then there may be some other more systemic side effects such as fever or headache or muscle pain. And these side effects range in severity from very mild to moderate, or in rare cases, even severe. But these responses are, for most people, completely normal. They are natural signs that your immune system is at work and your body is responding and developing an immune response, and antibody production is underway. However, that being said, if you are ever at all concerned about the side effects that you or a loved one are experiencing after receiving a vaccine, you should definitely contact your physician to touch base with them about that. So, given all of this information about how the virus works, how our immune system works, and how the vaccines are effective in inducing an immune response, there are a lot of questions out there about efficacy and how long the vaccine lasts and how much protection we have against viral variants. First of all, I think it's really important to point out that these vaccines are exquisitely effective. The Pfizer vaccine was shown to be about 95% effective at preventing infection after 2 doses. Moderna was 94% effective. The Johnson & Johnson vaccine was roughly 66% effective. These are really astonishing numbers. And we know that vaccination dramatically decreases your risk of becoming seriously ill or dying from COVID-19. We know that an unvaccinated person is 11 times more likely to die from COVID than a vaccinated person. And a person who is or hospitalization rates for unvaccinated people can be up to 31 times higher than for vaccinated people. And it is important that we work on vaccinating as many people as we can, because there are still people in our population who cannot be vaccinated, possibly due to a pre-existing condition or to an autoimmune disorder. Similarly, children, people under the age of 12, are not yet approved to receive the vaccine. And so the best way to protect these people, to protect this at-risk population, is to ensure that everyone around them is vaccinated. Because by doing that, we sort of ensure that the at-risk population is sort of in a protective bubble where everyone around them has been vaccinated and is therefore less likely to transmit the disease to them. There is pretty strong and growing evidence at this point to suggest that vaccination is likely to help prevent the spread of the virus. So vaccinated people who become infected are far less likely to actually transmit the disease to somebody else. This is because if you are vaccinated, you have these antibodies against the virus. So if you are exposed and unfortunately develop an infection, the viral particles that you have in your body are going to be covered in these anti-spike protein antibodies. So the antibodies covering the surface of the virus likely help to prevent infection in other individuals. And this is something that we are still studying, trying to understand better how likely is a vaccinated person to transmit the disease to somebody else. But mounting evidence is again pointing towards a dramatic decrease in the risk of spreading the disease if you yourself are vaccinated. But we do need to acknowledge that much like anything else, the vaccines are not perfect. Increasingly, we are hearing about breakthrough infections, and these are infections in people who have been fully vaccinated. These breakthrough cases tend to be asymptomatic or mild, and this is because the body has already been prepared with an immune response, right? You already have antibodies and can respond rapidly. And there are a lot of factors that go into the question of why people get breakthrough infections, far too many for us to be able to pinpoint any single cause for an individual. Things that might go into this that might matter in terms of trying to understand breakthrough infections include what vaccine did the person initially receive and how long ago did they get it? What is the fitness of that person's immune system? Do they have any pre or coexisting conditions? Are they old? Are they young? Are they immune compromised? What viral load or concentration of virus were they exposed to? All of these things may factor into trying to understand breakthrough infections better. As of now, there is some evidence to suggest that the Mrna vaccine is slightly more effective than the Pfizer vaccine in preventing breakthrough infections, but it's worth noting that even with this decline in vaccine efficacy that we are beginning to see a little bit of, both of these vaccines are still incredibly effective. A recent paper put Moderna at about 86% effective in preventing breakthrough infections and Pfizer at about 76%. And there is always going to be a range there depending on the population that was studied and when they received their shots, et cetera. But both of these vaccines are still incredibly effective at preventing breakthrough infection. And as we think about these breakthrough infections and more important, and how to avoid them, one question that sort of naturally arises is how long does the immune response last, right? Once you get the vaccine, how long is it good for? And here, preliminary evidence seems to suggest that the vaccine remains pretty effective and the efficacy remains pretty stable for about 5 to 7 months and then begins to decline after about 7 months. And this is part of the reason that the FDA approved a third shot or a booster shot for the Pfizer vaccine in September. And the idea here is to give a third shot to individuals 6 months after their second shot. And this provides a stimulus or a boost to the immune system to sort of restart antibody protection. Just yesterday, an advisory panel to the FDA voted unanimously 19-0 in favor of emergency use authorization of a booster for the Mrna vaccine. So we will probably see a booster for Moderna very soon becoming broadly available. And the thinking here again is that this third shot might add a little bit of extra oomph to the immune system and restart any antibody protection that had fallen off. Given the approval of the Pfizer booster and the likely imminent approval of a Moderna booster, there is an interesting question about mixing vaccines, right? If you received Pfizer for your first 2 shots, might it make sense, given the slightly higher efficacy of Moderna, to get a Moderna booster? Or given that as of today, only the Pfizer booster is approved, why not get the Pfizer booster regardless of which shot you've received previously? And currently the recommendation from the CDC is really that you stick with the same manufacturer, right? So only people who received 2 Pfizer shots could get a Pfizer booster. But that being said, there is mounting evidence to suggest that mixing vaccines is highly effective. A recent study looked at antibody levels produced when an individual received an initial shot from Johnson & Johnson, and then a booster that was either another Johnson & Johnson shot or a Moderna shot or a Pfizer shot. And the study found a significant increase in antibody levels when individuals received an initial shot from J&J and then a booster from Moderna or Pfizer. And for those people in the study who were initially vaccinated with an mRNA vaccine, either Moderna or Pfizer, they produced similar levels of antibodies following a booster, regardless of how the vaccines were mixed, right? So if people received 2 Modernas and then a Pfizer booster, that was pretty much the same as receiving 2 Modernas and then a Moderna booster. And so this is something that the FDA is actually hopefully going to discuss today. Today, the FDA is reviewing evidence in support of a J&J booster, the Johnson & Johnson booster shot. Part of the data that they are going to be looking at today also includes results from the study that I just mentioned, which looked at mixing different types of vaccines together. So they will also hopefully discuss the efficacy of mixing vaccines for the booster. It is definitely possible that they may focus their attention on trying to get boosters approved across the board first, right? Have Pfizer approved, have Moderna approved, have Johnson & Johnson approved, and focus on the idea of mixing vaccines later on. But there are mounting data that suggests that this is an effective strategy and may be a really interesting route moving forward. Another thing that we read a lot about in the news are viral variants. So what does this mean? Well, viruses mutate. What that means is that there can be slight changes to the structure of the virus that accumulate over time. And these mutations may result in a virus that is more or less infectious, more or less lethal. And generally, the virus mutations that take hold and spread are ones that make the virus more transmissible or more infectious. Because these mutations change the structure of viral proteins, this can pose a problem to natural immunity or to vaccine efficacy. Remember that antibodies are highly specific to a particular antigen. And in the case of SARS-CoV-2, that antigen is the viral spike protein. There is a possibility that the virus mutates in such a way that the structure of that viral spike protein changes. And if the structure of that viral spike protein changes too much, then the antibodies generated during a natural infection or in response to vaccination may not recognize this new mutated viral spike protein. That is a possibility. The good news is that currently we haven't seen a coronavirus variant that is completely able to evade existing immune protection. The Delta variant, which is currently the most common variant circulating in the United States and in many places worldwide, is about twice as contagious or transmissible as previous versions of the coronavirus, and it is more capable of causing severe disease. But even with that, the vaccines remain highly effective. We're running into a bit of a tricky phase of research now where there are multiple factors that are influencing data about vaccine efficacy, specifically against the Delta variant. On the one hand, we do have prevalence of this much more transmissible variant, but we also are reaching a time point where it has been for many individuals more than 5 to 6 months since they received their initial vaccine. So we are dealing with sort of this two-pronged problem of a more transmissible version of the virus and what may be slightly waning immunity in the general population. So it's tricky to precisely nail down exactly what might be causing an increase in COVID cases, particularly in vaccinated people. So I want to leave some time to answer questions that people submitted and any questions that anybody might have sort of on the call. But the take-home message, which I hope that I have made clear, is that the vaccines that we have currently for SARS-CoV-2 are safe. They are astonishingly effective. They are the culmination of years of scientific inquiry, and they really are a truly remarkable testament to international collaboration, and they really are our best path forward through this pandemic. We will continue to learn more. We will continue to adapt in response to new information. We will continue to seek out expert opinions and to perform well-controlled and careful clinical trials. And hopefully our ability to prevent and respond to SARS-CoV-2 and COVID-19 will only improve from here. Thank you all very much for attending and for listening. And I think that we are now going to look at some of the questions that may have come in over the course of the talk. So one of the questions that I see is how long these booster shots might be effective for. And this is one of those areas where unfortunately we just don't have data yet, right? The booster shots for Pfizer were just approved in September. So we don't have enough information yet to know how long they're going to be effective for. So we're going to need to wait and gather more information to see how long we have that sort of sustained and robust antibody response for. Let's see. Let's see.
Zoe, I had a question actually,
Yes.
while we wait and see if anyone else has other questions. My question is, do you think we'll see other vaccine types, or, we've got Pfizer, Johnson & Johnson's, Moderna. I know there are others that are being used in other parts of the world. Do you think there will be any other options here in the US?
I think that that's definitely something that will be considered. There are a lot of different sort of aspects of vaccines that make them more, maybe easier would be a better word. So the Johnson & Johnson vaccine, one of the major benefits of it is that it's a single shot and it's really easy to store, right? And so the more breadth that we can get in terms of the availability of vaccines, the more we'll be able to adapt and provide equity in terms of who is able to get a vaccine, right? How can we ensure that we are getting shots into as many people as possible? So I think that we will start to look at some more of the vaccines that are out there. The UK has AstraZeneca, we have Sputnik as well. So there are other options that will begin to be assessed once we have sort of our vaccines approved in the United States, we feel comfortable and confident that we are vaccinating our population and people are getting their first shots and their second shots. I think we will move towards a regulatory process for other ones as well. We will still put any vaccine that is approved in another country through our own clinical testing to ensure that it meets the standards that we have here in the United States for safety and efficacy.
Excellent, thank you. And now the questions are really rolling in. So there's a question, what do you think will happen with vaccines for younger children, which is a great question.
So that is a great question. I think that the looking at how COVID-19 impacts children is really interesting because COVID-19 actually has a pretty rare profile in terms of who is hardest hit. Usually when we see viral diseases like this, we see that young people and old people are most severely impacted. So children and people over the age of 65. For COVID-19, we have seen that really significant age skew where there's a really significant impact on people over the age of 65. However, there's very little impact on children. And so broadly, children under the age of 12, we've not really seen that much of an impact on them. So there is a really interesting question about vaccinating children under the age of 12. And so because children are not very likely to develop severe disease, and additionally, they seem to not be very likely to transmit amongst themselves, amongst other children as well. There are some people who are suggesting that maybe we don't need to be focusing on vaccinating children, right? Maybe what we should be doing is focusing on vaccinating adults, ensuring that all of the adults surrounding children are vaccinated, and then we can look at approval of vaccines for young children. So it is a sort of interesting question because we are looking at trials currently of vaccines for children under the age of 12. Right now, that mainly means adjusting dosages, right? Trying to figure out what would be an appropriate dose to give to a child, and how do we ensure a robust response in children. But given how mild and rare the disease is in kids under the age of 12, there is a sort of interesting sort of push and pull with the question. And so it's definitely on the minds of a lot of people. I could say that from a personal standpoint, I have a 15-month-old, and if he is eligible for a vaccine, I will sign him up for one. If that becomes a possibility, I will excitedly take him to receive his first mRNA vaccine, but that was my personal decision. And other people may feel differently about the vaccine for their kids.
Excellent, thank you, Zoe. We got many questions now, so I don't know if you want to take a look and see which one you want to answer. There's one about travel.
Let's see. So there is actually a new drug. So that's an interesting question. Yeah, so there was recently some data released that showed a pretty significant impact of a new drug in preventing severe infection or hospitalization in people who have COVID. And so currently we have drugs, we have remdesivir, which has been formally approved in the treatment of COVID-19. Remdesivir has, unfortunately, as we've gathered more data about it, has not been shown to be incredibly effective. It's a good drug, and it works to some extent. But there is a new, there were interim results released from this trial that showed that there was a new drug that was much more effective in preventing severe disease or death. So in the trial, they looked at, memory serves, roughly 700 people, and they gave them this new drug compared to people who did not receive it. And there was a marked increase in the rate of death and hospitalization in the people who did not receive this new drug. So the results were actually so striking in this trial that they stopped the clinical trial because it was considered such a sort of overwhelming success, demonstrating how effective this new drug is likely to be, that they wanted to push through to the next stage of approval to try to get the drug's authorization to be used as quickly as possible. So the name of the drug is completely escaping me in this present moment, but it is a really exciting possibility. And so I think it is something that we will be hearing more about soon.
Excellent. All right. There's a question about how likely do you think we will see further mutations of virus if a significant percentage of the population does not get vaccinated against current variants?
So viruses mutate rapidly. They mutate pretty consistently. We have seen that these mutated versions of the virus, they sort of need to hit this sweet spot where they are able to increase transmission, so increase how contagious that version of the virus is, and are more easily able to be spread in between people. And the way that the virus picks up all of these mutations is through reproducing and through going through more iterations of infection. And so the more widely a virus is able to spread, the more quickly we are likely to see it mutate. So if we are able to try to slow down or prevent the spread of the virus, we will hopefully be able to diminish any potential impacts of mutation. So hopefully, again, we will continue to vaccinate more of the population, and that will enable us to begin to slow the spread of the disease. And if we can slow the spread, then we will also slow the rate at which it is mutating, and we will decrease the likelihood of having one of these new variants take hold and spread.
Thank you. Here's another really good question about the FDA. So the current vaccines are emergency use authorized. Can you explain that status, and when will they be approved under more, quote, normal approval standards?
Yes, so emergency use authorization is effectively something that says, we desperately need this. We need this immediately. We need this to be available to the general population as quickly as possible. I think it's really important to note that that does not mean that it has not been through the same rigorous testing standards as something else, right? We still go through all of the preliminary safety studies where we look to see, is the vaccine safe, right? Or is the drug to treat COVID-19, is it safe? Before we even begin to look to see, is it effective, we look to see, is it safe, right? We've still done all of those studies. Then we begin to look at efficacy, right? And we begin to look at, is it effective in preventing infection with coronavirus? Is it effective in preventing hospitalization and preventing death? And once we have answers to those questions, once we see what the safety and efficacy profile looks like, what the emergency use authorization does is it allows us to very rapidly push through and begin manufacturing and getting the drug or the vaccine into the general population. So we will likely see all of these things go through and be formally approved by the FDA and enter into what people generally call phase 4 or post-market surveillance, which is when we're just sort of keeping an eye on the general population and still ensuring that the vaccine or drug is meeting safety and efficacy standards. We will definitely work on that. And I think that Pfizer and Moderna and Johnson & Johnson are working towards full regulatory approval of all doses of the shots. The reason at this point that we're working with this emergency use authorization is just that it's faster. And unfortunately, getting a drug or a vaccine approved involves, to some extent, a lot of paperwork. And we can get things done faster if we have an emergency use authorization and we are administering while we're working out all the sort of formalities of full approval.
Thanks, Zoe. Knowing that it's not your area of expertise, but perhaps you can give your opinion, do you see travel going back to normal, or will you be required to get the vaccine in order to travel?
So that's a really interesting question. And I think that will probably be left up to individual countries to decide, right? And that will probably be a decision that, an individual country or even possibly, depending on where you're going, sort of an independent region, right? Like an airport might be this sort of guard post for entry into a country or entry into a region of a country. And so some countries may choose that they are only interested in receiving vaccinated travelers, right? Some countries may choose to decide that they are interested in receiving anyone. And my assumption would be that countries that are more heavily dependent on tourism would be possibly more likely to sort of allow anyone in. But again, that might not be true across the board. I would think that to travel, you might want to make sure that you're vaccinated, just because, again, any time that you travel to a new region, you do run the risk of maybe being exposed to a variant of the virus that you would not have been exposed to had you stayed in Massachusetts or in the United States. And so there is some level of increased risk that will come with travel, also airplanes, but yeah.
Thank you. Another question about mRNA vaccines. Do you see any possibility that they could be administered through mediums other than a shot? This question goes on to say, if there continue to be new variants, it may become increasingly burdensome on the healthcare system in the US to regularly update vaccines. If vaccinations could be administered through a pill, for example, new vaccines to new variants could be administered more quickly. What are your thoughts on such a solution?
True. So the delivery of vaccines is something that we're really interested in, and I do think that it's worth noting that we are pretty good at adapting to new variants of viruses. Right. This is why there's a new flu vaccine every year, because every year a panelist of experts makes a call about what are going to be the predominant variants of the influenza virus that year, and then we develop every year a new vaccine for that particular single variant or collection of variants that we think is going to be of concern for that flu season. So we are able to do this, right? We do have a sort of manufacturing base that's already in place for annually or more regularly updating the variants of a vaccine that we are targeting. So we might end up in that space, right, where we are receiving updated shots, maybe every year, maybe every 6 months, that will protect us against a new variant of concern or whatever variant is likely to be predominant in that region in that year. The question of how vaccines are administered is really interesting. And for the mRNA viruses, for the mRNA vaccines, and also for a lot of the vaccines that we already have, a pill form would be really difficult. The reason that would be really difficult is because when you take a pill, it's going to go to your stomach, right? And your stomach is a really harsh environment. It's very acidic. Its job is to break things down. And DNA and RNA are fragile. RNA is very fragile. And so it would be very difficult to get the RNA to remain stable in that incredibly harsh environment. So we would be really reliant on that sort of protective coating on that lipid particle, or in the case of J&J, on that adenovirus vector to protect the contents. But then it would be very difficult to actually get the vaccine where you want it. So I would say that the idea of a pill would be challenging. There are some forms of vaccines that children receive orally, so there's some history there, right? And mRNA vaccines are new and formulation may advance, so we might get there. One version of vaccine delivery that people have also been really interested in is intranasal delivery, where it's effectively sort of shot up your nose. And so that is another avenue that people might explore. The ability to deliver vaccines in these new ways is predominantly what's called formulation. If you think about a drug, right? If you just take Advil that you pick up at a pharmacy, you have an active ingredient, and then you have that incredibly long list of inactive ingredients. Those inactive ingredients are the formulation. They are all of the other components of that drug that are necessary to ensure that your single active ingredient, ibuprofen or whatever it is able to get where it needs to go and be effective. And so all of those components of the formulation are what ensures that the vaccine or the drug stays stable, that it isn't contaminated, that it's able to get where it wants to be. So it is possible that we will work out a formulation for these vaccines that allow intranasal delivery or that allow maybe oral delivery. That would be very exciting. But we don't have oral vaccines for adults very commonly at this point. So intranasal might be a slightly more realistic goal. But we would see that both would be really exciting, right? That would be an amazing advance if we were able to do that. So fingers crossed that people are working on it. But I do think it's important to point out that we do have the manufacturing capability in place to continue to really just churn out shots. A little bit unfortunate to get, but that might be where we are.
Thanks, Zoe. We have time for one more question, and there is a question about, have there been any long-term effects found, and side effects, I'm assuming, that have been found with mRNA vaccines? And there's a question specifically around reproductive issues.
Yeah, so we at this point don't really have long-term data, right? So when we think of long-term data, realistically, we probably want years worth of data in order to make any sort of significant call there. And unfortunately, we don't have that yet. We have, for some people, maybe a year out from when this vaccination campaign first began in large numbers. So we don't have any information yet about long-term health effects. I do think it's worth pointing out that there are no vaccines currently administered that do have long-term health impacts, right? So it would be very unlikely for there to be any sort of long-term health impact. And specifically when it comes to questions of fertility, this is something that has been sort of perpetuated a little bit. And we really don't have any evidence that there's any reason that these vaccines would cause an impact on reproduction or fertility in any way. In fact, there would actually be, there's some evidence of a pretty significant benefit that if a pregnant woman receives the COVID-19 vaccine, we have found antibodies in the child, right, in the fetus. And so what that means is that as the fetus is growing, the antibodies that the mother produces against the virus are passed on to the developing fetus. And so that child would be born with some level of protection against the coronavirus. And again, these are sort of incoming data. We don't know exactly how much protection that might be, but the evidence that we have so far actually points towards there being a benefit to receiving the vaccine for safety in infants.
Excellent. That actually answered one of our other outstanding questions. So look at that, 2 for 1. Well, thank you so much for your time. We will be sending out a recording of today's session as well as Zoe's contact information. So please feel free to reach out with other questions that she might be able to answer for you. Thank you so much, Zoe. It was a pleasure as always to have you back talking about viruses and vaccines, and hopefully everybody stays healthy.
Thank you so much. Bye.
Good afternoon. Bye-bye.